Next is Reactome database. So all these are part of annotation. Now earlier we have checked that the proteins which are involved in different gene ontology category, that may be biological processes, molecular functions, or cytoplasmic location. Now we're interested to see that those proteins are involved in which biological pathways. Now pathway information, why this is important? You know that very well. For genetic engineering purpose, suppose you want to modify, or um, you want to what I say, uh, overexpress some genes in a cell, in a bacterial cell. Okay, so then you may look into the pathways of that product production, right? Which by which pathway that product is processed. So the and on which enzymes are key uh, enzymes in those pathways. If we can increase those enzymatic activities, so easily we can modulate that pathway, and we can uh, express that proteins in larger amount in a cell, in a bacterial cell. So in that perspective, pathways are very much important. And these pathways, how these pathways are developed, because only one group of scientists cannot invent one pathway. That is very basic concept, isn't it? Because there are lots of molecules, lots of enzymes are involved in a pathway. If you think about that from simple pathway, that glycolysis, so it has 10 different reactions, 10 different enzymes are associated with. So, so many scientists are working for a particular mm, problem solving, and they are trying to identifying some what I say, substrate and product relationship. And after that, someone can connect the dot. If someone can connect the dots, so then he or she can invent the full pathway. From the biochemical perspective, you can understand this thing. So, uh, what are the challenges in this pathway? That means that the information is spread across many several hundreds of papers. So, as I have already mentioned, that we have discussed, we have uh, studied many different things in many different research, and those are published in different articles. So from those different articles, we need to gather the information first, and then we need to make the connections. This is a very challenging task. Often this kind of scientific exploration are ongoing. That means this is never ending process. This will be updated soon. So even if you have identified a pathway inside a cell, that pathway may be changed, may be modified, may be updated. Okay, if you go back to 50 years ago, the biochemistry book they used, so there the pathway concept, and nowadays the in the textbook the pathway concept, you will find that some modification, some updatation has been performed. Why this update has been occurred? This is simple fact that the proteins which are present in a particular condition, and those are not same, those are different. And most importantly, due to the advancement of the techniques, tools and techniques, we can find out many novel proteins inside the cells, and those novel proteins can be involved in the pathways also, the previously identified pathways. Okay, so that's why, and the protein annotations are also being modified. So that's why this is a mm, challenging task to design the pathway data. And it is not easy to simultaneously investigate different types of biological information in pathways. That is also another level of challenge, that in a pathway there might be different levels of things. That means the positive regulation, the negative regulation, the feed forward loop, feedback loop. Okay, so this kind of regulations are also going on, and it is not accessible for computational reuse also. So pathways, pathways, pathway databases are need to be updated regularly. Otherwise, that is of no use. So Reactome can help in this regard. These this is based on the computational data mining, and this kind of data can be reused. So it is a curated, quality assured slice of pathway information. These are this can be visualized as a diagram. Okay, and you can download these pathways also as a figure. So you can see that inside the cell in this diagram, it is showing that from these thousands of paper, scientists first develop this kind of pictorial views. You have seen this kind of cartoons in your textbooks also, and from that cartoons, scientists have developed this kind of pathway. That means one protein is how one protein is related to another protein. Which one is substrate? Which one is product? Kind of thing. Okay, so this is the way how Reactome database is evolved. It is a free database. Free means you don't have to pay money for it, but uh, free doesn't mean that it is not regulated. It is not uh, organized. It is well organized, and there are several people associated with it who are monitoring these changes time to time. They are peer reviewing these processes, and they are helping to curate this thing knowledge base. Okay, and provide a user friendly platform to access and analyze the information on these molecular mechanisms of reactions and pathways. You can understand, or you can remember the fact that not only you can browse this thing, you can download the data also. That is very important for research purpose. Okay, so you can download the whole website database in standard format, for example SBML. So this is the format, systems biology markup language. Similarly, another one is biopath. So these are different formats, just like HTML, XML. This is also some formats which are particularly used for biological data handling. Okay, so another interesting what I say research wing is that how we can analyze, how we can develop a unifying platform for studying this kind of large scale data, because data analysis is not very tough job using different tools, different software, but the curation of data is very difficult. When you will try to map map different types of data, then you will face that several kinds of problems. Suppose you want to identify which proteins are present in gene ontology database and which proteins are present in the reactome database. Okay, then you will find that there are huge difference. Some proteins are present in Go database, but those are not found in Reactome database. There are many different reasons for that, and that's why this kind of analysis are not very easy. Are you getting my point? What I am trying to say? Where are the limitations? It is not easy to get simple solution. So research must go on. Whenever you will find some network module and then that module you want to identify, Often you will find that using Reactome database, you are not getting any output. Using Go ontology, you are not getting any output. So you need to rethink that where is the fault, where is the error, why these things are not there. That kind of question will arise. So ultimately, you will need more research on that, and that's how the research progresses. Okay. Next is some terminologies with this reactome. That is the reactions. You know that basic unit of this database is a reaction, which is defined by any molecular event, including the binding, phosphorylation, or biochemical catalysis, molecular transport, or spontaneous molecular event. What is pathway? Pathway is defined as a series of connected reactions. So each individual reaction considered as a single step in the pathway. So these pathways are represented as a diagram. We have seen in many different processes like signaling or cell cycle or apoptosis. So in Reactome, you can use this, you can identify this kind of pathways and you can download that pathway data sets also. The gene or the proteins which are involved in any kind of pathway. Next, we can also curate this database manually or experimentally verified with the manually or experimentally verified databases, and this database is updated. Okay, often quarterly this database is being updated. Okay, so there are lots of tools actually. Then next, all right. So these are very common to you, that what is biological pathway, how it looks like, uh, why are pathways significant from the biological perspective. These are all known to you. We can compare the healthy cell versus the disease cell, and we can compare the biological pathways in that way. And obviously, the faulty pathways can result in diseases such as cancer or diabetes, etc. And interestingly, some medicines can correct these faulty pathways, and often by switching a signaling pathway on or off. That is very important, because you can understand that most of the drugs are acting on the GPCR, that is G protein coupled receptors, and how this kind of pathways mm, are analyzed inside the cell, that is also important, because if you can try to identify the targets by experimental tools and techniques, it will take lots of time. Only one pathway you may need 5 to 10 years or so, more than that. But if you use this kind of computational tools and techniques, you can find out a solution within a year or a couple of years. So that is the advantage of computational biology. Okay. So reaction pathway steps. These are the reactions of the units of this pathway databases. So many different types of reactions are represented by different formats like this. The binding, dissociation, degradation with this arrow. So from this molecule, this is prepared, this is synthesized, so degradation. Similarly, phosphorylation, dephosphorylation, classic biochemical pathways, and the disease. That means this is the inhibition symbol. That means one protein is mutated, and due to that, some diseases can occur. This is the way how diffusion is represented, and this is how the transport is depicted in a diagram. So these are the different notations you can see in a database. Okay. Similarly, here reactions connect to form pathways. That means here one substrate is forming a product. That product again for the substrate of reaction, and that is again forming another product, which is a catalyst here, and that is regulating another reaction, and one substrate is being converted to a product. Okay, so many different types of relationship may exist in a reaction pathway, that you should remember. So when I was working on my rice microRNA mediated pathway analysis, so I found that some enzymes which are involved in inter pathway, and some are involved in many different pathways. So intra pathway and inter pathway. Okay, so many types of relationships are there. So we can analyze the whole database, and then only we can get the information. We can identify some information about the regulation inside the pathway. Okay. Those are very basic things. I don't need to repeat this thing, how these data are curated. So after the experiment have been performed, some curators are there, some data analysts are there who are actually building the database, and that after the completion of database creation, that will be reviewed by some scientists, and then only it will be published. Okay, the Reactome is updated quarterly. It is mentioned here. So it is also important to update some database, existing database. Often you will find that many databases are not being updated 1 year or so, or 2 years or so. So that kind of database, if you are using in your research purpose, so there will be some problems in future. So you should find out the updated database, and you should use that data. So you can see that it is a very old, not very old, but again, almost 20 years ago, this kind of database has been developed. And 2020 data is showing that around 2,500 human pathways and more than 13,000 reactions, 10,000 proteins have been incorporated in this large database. Okay. These are the cross references. So since this is a relational database, you can, they are using different databases data, linked data like UniProt, EMBL, PubMed, RNA Central, Go, and CHEBI. So these are the cross references. Okay. Next. Yeah, that is very important. This thing, that how this kind of database analysis may help to manually infer the pathway. So look at the example carefully. Suppose most of the experimental work have been done on human pathway, isn't it? So from different literatures, you may get that there is a pathway exists in this way. So pathway 1, pathway 2, pathway 3, and pathway reaction 1, reaction 2, reaction 3, reaction 4. Now, from mouse or from cow, you may get that this reaction, this same reaction is going on in case of mouse. Similarly, in cow, this same reaction is going on. So these are the indirect evidence that maybe this reaction is controlled or regulated with the same group of molecules which are present in the human pathway. Okay, so this is another way, another method how you can infer the pathways in many different organisms also from comparative analysis. Is it clear? Any question? Okay. If you don't have any questions, so then next one is beside the manually curation. So computer can be used for the same purpose, and here you can see that three different reactions is shown, for one for human, one for mouse, and one for Drosophila. And the same reaction is going on. A plus ATP, which is catalyzed by B and is forming AP, and then plus ADP. So the same reaction is going on in mouse and same reaction is going on Drosophila. But when we are projecting this thing, we found that uh, human and mouse both have all these molecules present in that pathway, and they are exactly uh, controlling the same set of reactions. But in case of Drosophila, this molecule is not found, but B is there. So ultimately, what will happen? Reaction will be absent. This set of reaction will be absent. So you can infer that B, if it is present in this Drosophila, so that is involved in any other function, not in this function. That is very much obvious. Okay. So this kind of computational prediction you can do by using the data. Okay. Next, this is the important section, that when to use Reactome. So some are very important points, like understand the comprehensive molecular details of pathway based on available literature. That is very obvious that from the literature you are going to develop some pathway, and it will help to know how various proteins and small molecules interact in vivo to achieve biological function. From the molecular perspective, you can also derive the biological functions. The third topic is very important. It will help to identify the crosstalk between different pathways. And inside the cell, this happens, crosstalk. That means in a pathway, suppose some glycolysis is going on and then Krebs cycle is going on. So those are two different pathways. Now, if you are thinking that where is the link, which proteins or which reactions are shared in between two pathways, if you can identify that thing, then that examples, that kind of examples will be termed as crosstalk in between the pathways. Am I clear?
Yes.
All right. And when not to use reaction? There is also another question. That as the reactions data are incorporated in the Reactome data, obviously, when you are interested to find out protein-protein interaction data, you will not going to use Reactome data. Okay, so it is very common fact. For interaction prediction or interaction data, you can use many different databases like Intact, DIP, etc., etc. Okay. Now, very common fact that why this kind of reactome are very useful. We have already mentioned that from the high throughput experiment, uh, we can analyze the data, and we can identify that which genes are upregulated or which genes are downregulated, and what are the uh, functions of these upregulated or downregulated gene sets. So for that, we can map those proteins or those genes to the pathways, and we can derive the pathway specific property. Okay, so that is the way how it is working. Okay, okay, one thing. As a bioinformatician, you should follow this thing, because how this kind of databases are stored. This Reactome data is stored in a Neo4j graph database. It's a type of database which is using Neo4j algorithm, Neo4j graph tool. And this is allowing the cipher queries and a MySQL relational database with a fully defined schema. So it is a relational database and which is based on MySQL. I think you have heard about this query language. And Reactome is also included in GitHub now, and this data can be downloaded in several standard formats. Now again, I am not going to discuss about the navigating Reactomes and understanding the pathway browser, because all these are very much practical lab oriented work. If we have some time, so then we will do this kind of analysis in lab. But here in this section, it is described that what are the different analysis we can do using the Reactome database. That is pathway enrichment analysis, gene set enrichment analysis, molecular interactions overlay, expression overlay, disease, species projection, tissue projection, etc. Functional interaction analysis also. We can use the data, we can access the data programmatically, because we know that how the database is created, and we can download the data, and we can use the REST API based service for programmatic access. Okay, so this is another added advantage, that if we write a program, and if we are using this, if we are trying to get the data from that database using some program, that is also possible. Okay, that is not possible in many other databases, but nowadays this kind of programmatic accessibility are added to this kind of large datasets, which will be useful for future generation. Okay. If you are interested to contribute to Reactome, you can contact them. There are different systems for doing that. You can directly write to help@reactome.org, and it is uh, they will review your suggestions, and accordingly they will update the database. Okay, so this is the thing. If you have any question, you can ask.